So thank you so much for letting me talk here. It's I've been following this seminar series since the days of COVID, and it's been going from strength to strength, maybe until today. um, Also, thank you, David, for the music. It was by a Dutch composer. There's not many Dutch composers, so great that you found some. If I were to choose the music, I might have taken this CD. This is The Art of the Cigar by the famous Belgian director Paul van Nevel. And it's all songs and about cigars because both Carl and I, we like our cigars. And one of my best memories with Carl was that we were at a conference in Germany somewhere, and we came back to the hotel after a couple of drinks, and we decided to smoke a cigar because I had some with me, and we couldn't smoke in the room, of course, so we had to stumble on the kind of fire escape, and I'm afraid we woke up some people. If you were one of those, we're very sorry, but it was very enjoyable as well. Okay, let me get started to what I want to talk about today. As you probably have heard a couple of times, that the brain is quite a metabolically intense organ, consumes about 20 watts, about the same as a dim light bulb, so to say. And this energy is really needed to restore mostly ionic balances, because as soon as we have an action potential or synaptic transmission, there's flow from ions across the membrane, and these ions have to be restored, and that means pumping often against the electrical and concentration gradients. So Atwell and David Atwell and Simon Laughlin came up with this famous estimate then that about half of the energy goes towards synaptic transmission, about a third goes to spikes, and there's some other costs as well. And really thinking about the brain as a device that needs to be careful with spending energy, using this as a design principle explains quite a number of things about the brain. For instance, why would we use sparse activity? Why is synaptic release so unreliable? Why is the potassium current shaped as it is? And these can be kind of helped to explain by thinking about energy. There's also a suggestion that the rise in the brain size in humans coincided with kind of the invention of fire. So at that point, humans could cook food, and maybe they had then more energy available for their brains, basically. Also, currently, there are still ideas that availability of energy is important. There's a famous study here about judges in an appeal court, and they're very favorable unless you're right before the lunch break or for the tea break, because they then don't have much energy anymore to make a difficult decision, and the proportion of favorable decisions goes down. I mean, there's multiple interpretations possible to these things, of course, but this is one interpretation. Also, in children, maybe in particular, you find that having breakfast or not, this is a political issue in the UK, has actually effect on academic performance. So the ones that have breakfast, school-provided breakfast, perform better than the ones that don't. As I said, particularly during human development, this is important. If you look at the amount of energy used, this is all at rest, by the way, by the brain, is much stronger in children than it is in adults. Okay, that's all about, in a way, energy to compute, perhaps, but there's also energy needed to learn. And I first came across that in this nice study from Miri and Koretsky, which did the following experiment. They had a fly that was exposed to an odor, but whenever that odor was present, it also got a shock. And of course, these flies are not stupid. And if you repeat that 4 times, this association between the odor and the shock, then, and you ask it to make a decision, then it will stay away from that odor because it doesn't want the shock. Now, that was all known, but what they did in their experiment was kind of unique. They stopped feeding the flies after they've learned this task. And if you stop feeding them, they die. And this is indeed what they measured. So you see here the flies that learned the association in white, and there's some control conditions in yellow and red, where they basically didn't learn, there was no association to learn. And you see that the flies that were exposed to the protocol died about 4 hours quicker than other flies. And the interpretation is really that so much energy is needed to learn this association that if you stop feeding them, they die more quickly. There's other evidence that also after if you don't starve your flies, but are a bit nicer to them and keep feeding them, but measure their sucrose intake, you find it almost doubles after learning. And this is almost like children coming home from school hungry for anything sugary. So you can make very simple estimates how much energy this actually corresponds to. There's a beautiful machine, this calorimeter, which you can use for flies, and you can find that the fly metabolic rate is about 8 microwatts. We humans are at about 100 watts. And you can then make a coarse estimate, and you find that if there's just one bit of information to be learned, that's the association whether odor is good or bad, then it would take about 10 millijoules per bit. This seems like a very high estimate, but this is at least in that order of range we're thinking about at the moment. And you can compare that to other devices in your computer, for instance, and you see that biology is not particularly efficient. Now, this whole idea of energy and computation and learning is, of course, not unique to biology. Here is some newspaper article that Google just wants to buy a couple of nuclear plants, just 5 or 6 or 7, that should be enough. And large language models are, of course, notoriously expensive energetically. And hopefully, with new architectures, this will get better. I think in memory computing and neuromorphic architectures. And it might well be that these memory costs actually become more and more important. Now, as a computational neuroscientist, you might ask yourself, why is learning so expensive? There's a whole bunch of processes that gets put in motion as soon as you learn something. There's protein synthesis, there's transport, there's actin, there's anatomical changes, there may be replay activity, also in flies. And all these kind of processes can be estimated, but to be honest, we don't really know at the moment what takes all this energy. So what you can do is, as a computational person, you can just say, well, we make an energy model that is probably wrong, but we give the assumptions that we use. And once we get more information from biology and the experiments are close to underway, we can update our energy model. Our energy model is going to assume, first of all, that it's a synaptic plasticity, that it's costly. That is, in a way, a bit of a worst case scenario. We can discuss these things afterwards. And we also assume that there's no cooperative or competitive effects in the energy consumption. You can imagine that when you have to potentiate 2 synapses in parallel, this could either be competitive, it could be more costly, it could be less costly. I could think of either case. We don't have any data on that, so we just assume it's not present at the moment. So if we track weights, then we have to still say how much does it cost to change synaptic weight. And we came up with 2 models. It's kind of 2 reasonable things we thought. The first we call the M1 model. It's after the L1 norm, which just takes all the changes in the synaptic strengths for each individual synapse, sums that over all the synapses and over all the changes that it makes. And it doesn't matter if the synapse goes up and down in strength, it counts the same. You can make some differences there, it doesn't really matter. And so if on the right there, you see that the weights go up and down during learning, you would just calculate the length of all those red bars, add them together. That will be the energy. You can also take the L0 norm of this quantity. We call it the M0 energy, and that's just counting the number of updates you make. So that's, for instance, relevant if you think about computers. Doesn't matter how much they change, as long as they change, you pay a price. Now that we have this energy model, we can then kind of think about how much learning costs. And to do that, we done a couple of tasks, but the main task that I'm talking about in the beginning is this simple supervised learning, the well-known MNIST handwritten digit data set, which we have a network with one hidden layer with an arbitrary number of hidden units. And the objective of the network is to, when this digit is present, to have output number neuron 8 active and the other one silent. We train them always to 95% accuracy, so they have a kind of a like-for-like story. We use backprop. We don't care too much about the biology of that at the moment. And we don't use batching because we don't really see how that could be done biologically, but there's something to say about that later on. And we fix the learning rate. So how does that work? If you then train the network, well, you get your sample, you calculate the output, you calculate the error in the output, you calculate the gradients in your network, you update your synapses, and you calculate how much energy was needed to do so. And then you go to your next sample until you reach your criterion. The first thing you have to fix is the learning rate. We kind of choose an intermediate learning rate. We find that here you see the energy measure M0 and M1 as a function of the learning rate. You see the bottom plot, the M1 energy goes down as you reduce the learning rate, because the more careful you navigate your weight space, the lower your energy. The M0 energy, so the number of updates, has actually an optimum learning rate, somewhere sitting there at about 0.01. And that's the one we take for our purposes. So it's not the most ideal learning rate, but it's a fixed one and reasonable, rather than optimizing always. So giving those kind of assumptions, we can start now to think of the energy spent to learn this MNIST task and think about ways to save energy. The first thing we're going to do is to update only some synapses, and this is in particular important for biological vision. So if we change the network size, and by that I mean changing the number of hidden units in the network, the energy obviously goes up, right? Because there's more synapses, more neurons, and so we spend more energy. The number of updates, this M0 energy, scales more or less linearly in the network size. Slightly less than that because bigger networks learn a bit faster. You see also, if you make the network very small, then it actually can't learn it very well and the energy goes up again. So there's an optimum there. This M1 energy actually more or less increases like a square root in the network size. And that's because the larger the network, the smaller the updates become. That has to do with the smoothness of the cost landscape. As the network gets larger, the landscapes get smoother and the updates become smaller, even if the learning rate is the same. So a machine learner might take, therefore, not a very large network, but biology doesn't have that choice, right? If we ourselves or a macaque has to learn the MNIST task, it has to do it with its brain. And so if a macaque would learn MNIST, this is the amount of energy. So it would be spending way too much energy because it has such a large visual cortex to do so. So a pretty obvious solution then is to kind of sparsify the gradient. And the first thing we did actually is to just take a fixed mask and say, before we start any training, to dedicate some synapses to be plastic and other ones to be static. And if you do that, you find that this increase in the energies as a function of network size is strongly reduced. They become almost flat, independent of network size, right? So those are the blue curves. It turns out you can actually do a little bit better. This M1 energy, you still see it going up. You can actually use what's called coordinated masks. And that's to say, if the inputs to a neuron are plastic, then so are the outputs. And so we kind of form plastic paths through the network. And that's a little bit more efficient, takes you less, requires less of this energy. This is actually something that there's some links to machine learning here, in particular in large language models. You might have heard of this technique called LoRa, low rank updating, where you basically train your large language model to incorporate new information, and you do that with a low rank matrix. And so, for instance, this delta W there equation is just the outer product of two vectors in this case. So that's a rank 1 matrix. And that is exactly what you do when you coordinate the input and the output plasticity. Also in neuroscience, there are some related effects. There's an old paper from Min Pu and collaborators where if you induce plasticity between the two red neurons, there's a bit of leakage of plasticity actually to the input neurons. And they've recently put that into a model as well. So that saves energy. You can actually do a little bit better still by having a competitive scheme. So rather than saying, okay, some synapses are plastic and some are static, every update we have competition, and only the ones with the largest updates actually get expressed, the other ones get zeroed out. And that further reduces the energy that you need. And also that you can coordinate again. That's what you did in this slide. And that's the lowest energy we can achieve basically. And we make the learning independent of network size. So there's another advantage of this competition effect. First of all, it's of course very easy to imagine that there's some kind of competitive interaction and some thresholding going on in some biological cascades. Furthermore, if you have a fixed mask and you take your mask too large, so to say, too few plastic synapses, then your performance will go down. If you have, however, a competitive scheme, then you can still always reach the right performance. It might take a lot longer, but you will still have high performance. So that's one way to save energy. That is basically by spatial sparsification of the updates to your synaptic weights. The next way to save energy is actually to use it in the temporal domain and to learn only from mistakes. It's a temporal sparsification. So what am I talking about is the following. If we do traditional backprop and we're getting presented this digit over here and we say it's a 2, then obviously we made a mistake and we should apply backprop and we should change the synaptic weights. Now in traditional backprop, even if you say it's a 7, but you're not 100% sure, you say I'm 95% sure it's a 7, then you still update all your weights. And you could imagine, can't we skip that step to save energy? Indeed, you can, and that is work by Aaron here. And basically, we now only update the synapses if the network makes an error in the classification. And so if you compare the blue curves, that the traditional backprop network of different sizes, to the red curves with this gating, also for different network sizes, you find that always this gating network gives you more performance for the same amount of energy. But there's a little bit of an issue there, because you'll find that the maximum accuracy you can reach with these networks, if you do this error gating, is not as big. It's a bit hard to see, but it's easy to imagine that if you are close to the decision boundary, if you then slightly try a different sample, so you try a test set and you're close to this decision boundary, you start to make a mistake. So your learning is not as robust when you do this trick. So there's actually a way around that, and the way that we found works best is to not only learn if you make a mistake currently, but also if that particular sample led to a mistake in the past. So you keep kind of a list of samples that you did once wrong in the past, and then you can still reach the same accuracy. The algorithm is extremely simple, and so when people ask me, hey, does this also work for my learning scenario, then I say, well, you can easily try it. So just to remind you, if you have standard backprop, you get your prediction, you update your weights, here you get your prediction. If your prediction is not what you want it to be, then you say, this is a sample I have to remember. And then if the sample is remembered, if it's in your memory cache, so to say, then you update your weights. And this memory cache gets never reset. So all the samples that you ever did wrong in the past always lead to updating. That saves a substantial amount of energy, as you can imagine, because at the end of training, you're 95% correct or so, and you can skip over most samples. The larger your data set, the bigger the savings you have. There's actually, in addition to MNIST, there's also the eMNIST data set. That's the extended data set for MNIST. Has 4 times as many samples, and you can save more energy there because the samples are more redundant, so to say. So that's nice for neuroscientists, for machine learners. I think also for neuroscientists, there's some message here, because if we think of the hippocampus as the thing that remembers things during the day and then consolidates them at night, if you only need to remember the things that you did wrong during the day, it might still be a large list, but it's probably less than remembering everything. And so the number of things you have to keep in your hippocampal memory to consolidate is much smaller in this case. So we think this makes sense in a biological context. Also, if you zoom in more on the neural level, we know that plasticity is not something that plasticity is sparse temporally. So in the top graph is a paper where they try to induce plasticity twice in a row. If you do that within 30 minutes, within an hour or so, you can't get additional plasticity out of the synapse. You need to give it some rest for, let's say, 90 minutes or so, and only then you can potentiate your synapse again. So there's a certain dead time. And there's also an older paper here by Peterson and Hopfield, actually, where they show that rather than having continuous small upgrades in your synapse as you do plasticity, that's actually nothing happening for a long time, and then suddenly it makes a big jump. It's an elegant way of showing it. I won't go into the details here, but it's a nice paper. So that's another way to save energy. Yet the third way to save energy is actually to use multiple forms of plasticity. So you might know that I showed you this slide before. There's different types of memory in your computer, and the more persistent ones typically are more energy demanding. It's no different in biology. Also in biology, we have different types of LTP, for instance, early and late phase, and both lead to potentiation of the synapse. But if you induce early phase LTP by rapidly stimulating your neuron in succession, then this plasticity decays over time. However, if you space it out, which we call spaced learning, let's say with 10-minute intervals or even longer, it leads to kind of persistent LTP. And these are really 2 different plasticity pathways with maybe even different metabolic supplies. The same is true in the fly. I showed you this graph already, but I showed you only the left part. Now in the right part, they did the same experiment with using this MAS protocol. So this odor shock association, rather than waiting 10 minutes between the associations, they did it rapidly in succession. And what you should see now is that this white bar, this reduction in lifetime of the flies, is very comparable to the controls. In other words, there's almost no energy that was used for this learning. So that is to say then, these kind of persistent forms of memory, they are energy demanding. The more transient forms seem to come almost at no cost. This might be due to protein synthesis, but to be honest, we can't conclude that at the moment. In any case, that suggests a way to save energy during learning, because as your weights move up and down as you learn something, you could try to accumulate all these changes first in transient weights and then only once in a while consolidate that. So we call this actually synaptic caching. It requires that your synapse has a transient and a persistent form, and we can think of pre- and post-synaptic components, for instance. We don't know exactly. And whenever the transient weight reaches a certain threshold, we consolidate it and we spend energy in making it persistent. And here's then the energy that you require is the M1 energy. In the case of traditional backprop, that's the red line, and our synaptic caching solution, the black line. So saving about an order, a factor of 5 in the energy needed. And the green line is actually if you could postpone all the consolidation till the very last point in learning. So you accumulate everything until you get to the final point and only then consolidate. So that's kind of a minimum energy that you could ever reach. So that then shows you a bunch of ways to save this kind of putative plasticity energy. To summarize, we can only update some synapses, and that's a very good idea if you have an over-dimensioned network. You can only learn when you make a mistake or have made a mistake on something, and that works well for over-complete data sets. Or you could use multiple forms of plasticity, and that's useful when you have access to a kind of more transient, cheaper form of memory. There's even other ways to save energy, and you can make plasticity adaptive. And in the previous experiment that I showed you with these flies, what they did is they had well-fed flies, taught them the association, and then started to starve them, right? What you can also do is to starve them before you do the experiment. And of course, the flies are hungry then when you do the experiment. And it turns out when they're very hungry, you try to teach them this association, they simply don't use all forms of memory anymore. And this was nicely shown in this paper from Pierre-Yves Placet and Thomas Preat. It turns actually out there's in the flies, there's a set of dopamine neurons, these MP1 neurons, that are active when the fly is well fed, but they are silent when the fly is starved. And so they code whether or not the fly expresses this expensive long-term persistent memory. And now with the nice things of fly genetics, you can artificially activate these neurons. And when you do so, the fly thinks it's well fed, expresses long-term memory, but then dies earlier because it spends a lot of energy on this memory formation. And so that's shown on the very right bar plot over there. So in general, so this is actually a mechanism that's also present in mammals, it seems. So in mammals, there's a sensor for energy use called the AMPK sensor that gets activated if you go from ATP, ADP, AMP, right, the standard cascade. And the more AMP there's around, the more energy you spend recently. This activates this AMPK sensor, and when it's activated, inhibits long-term LTP. And there's interesting links there with things like ketogenic diets and epilepsy and the like. So how might this be useful? Well, we actually can think of a kind of neuroeconomic problem. You can wonder, should animals actually invest in long-term memory? So this fly is exposed to this kind of choice every day. Should it go to one type of flower that is actually leads to a shock, or should it go to the other flower? And if it's low on energy, it has basically a dilemma. It can choose, should I learn about the bad flower and maybe die of starvation because I spent energy on learning, or should I not learn it and die of the stimulus? So it's a bit a sad situation. It's maybe a bit comparable to whether you decide whether to go to university or not. In principle, that should pay off, right, in the long run in terms of your income. But if you're broke when you go there, then actually it might not be the right thing to do. So how do we trade off these two different things, dying from starvation versus dying from the stimulus? In order to do so, we use this, what's called the hazard framework. This hazard framework has been developed in the insurance industry to kind of calculate how much life insurance you have to pay. And basically, every time step, there's a certain hazard that you die from whatever cause. And the two causes that we consider here, this hazard comes from two things. Either the stimulus is hazardous and leads to a certain probability of killing the fly, or it can die from starvation. And we assume a certain relation between the energy reserve and the hazard to die there that's shown in this graph here. So if you're low on energy, you are likely to die from starvation. So we can wonder what happens with these two forms of learning, these kind of transient forms called arm in the fly and persistent forms, LTM on the right here. And so when you learn with arm on the left there, then you learn a bit, but the performance stays quite poor because the memory decays with every between the stimulation. However, your energy reserve remains high because you don't spend much on the learning, and your hazards remain about constant. If you learn with long-term memory, then your starvation hazard increases. That's the bottom right plot there, but your stimulus hazard decreases. So maybe you survive longer or not as long. So you can think of different trade-offs. And here we show actually the lifetime of a population of flies as a function of the stimulus hazard. So the black curve is when there's no learning at all. The arm memory is always, the transient memory is always better because it comes at no cost. And you see that only when the stimulus is very hazardous, that's the blue line, it's better to use this persistent form of memory. Otherwise, it's just wasting energy or things that are not very dangerous. And so you negatively affect the lifetime because you die from starvation in that case. It turns actually out you can make a kind of a mix of a kind of an adaptive scheme where you say we only use this persistent long-term memory when, first of all, the energy reserve is large, so you have enough energy to spend, and second, your reward prediction error is large. And we played around with these schemes and we optimized these parameters there, the CM and CR, and you get this green curve that is plotted there, and you see that this leads to the longest lifetime. So our interpretation is that the fly has probably developed this kind of optimal scheme. We're still thinking about what's the best optimal scheme in general circumstances. This is in a daily protocol. This is also interesting effects for kind of appetitive conditioning. So appetitive conditioning is the same kind of learning between a stimulus and a response, but in this case, when there's a food response, for instance, and you see interesting effects emerging. So for instance, when the stimulus is occurring every day and you know that a certain odor is associated to food, your memory gets enforced and you don't really need this long-term memory because the one day is enough to carry you over the transient decay. However, if the stimulus is much more rare and you only get the stimulus every 3 days, then this long-term memory becomes actually better. So there's interesting interactions showing up there. Okay, so that brings me actually already to the end of the talk. So just to summarize it then, I think I've shown you evidence from experiments that show, or at least suggest that learning is a metabolically costly process, and therefore evolutionary pressure has likely found ways to save that energy and be frugal, so to say, to learn something without spending too much energy. Really, our interest in this is twofold. First of all, I think if we want to understand learning in biology and we don't take this constraint into account, we might get not a good picture of what biology is trying to do. And so, for instance, you see a lot of theory papers that assume continuous plasticity, and maybe that is not really what's going on. It's maybe very carefully selected. The second thing is that these algorithms that we develop might also help to improve AI. This is always a kind of a difficult battle because the people who do AI, they have kind of different constraints a little bit, but nevertheless, some of these algorithms do seem to work, and we hope that we can help there a little bit as well. Okay, that's it for today. If you're interested in any of this work, you can just go to this Google Scholar page of my lab, and you can find the papers that are accompanied, or you can send me an email, of course. Thank you very much.